Welcome, dear dreamer, to Dreamtime Discoveries, where science gently lulls you into rest. Tonight, we wander in quiet wonder exploring tiny worlds, hidden codes, and ancient echoes that soften the mind. Please like this video if you feel the calm, share it with someone who craves gentle curiosity. Subscribe for more meditative science journeys, and let us know in the comments what you'd love to explore next. Now, let your breath slow, and let our journey begin. You drift into a landscape of time's soft shadows, where extinct creatures whisper in your ear. Ancient peptides lie dormant in fossil genes, waiting for rediscovery. AI becomes your guide through millennia, reading molecular voices long silenced. Each peptide carries a story of hope for healing in modern bodies. Woven from deep memory, you breathe in the hush of distant time, the frozen tundra, the silent bones beneath centuries of earth. In this quiet space, you become aware of ancient life woven into tiny strands of genetic code. These strands, peptides, are fragments of proteins once alive in creatures long extinct. You sense them not as relics, but as gently glowing echoes, offering traces of their original strength and purpose. Your journey begins with the woolly mammoth, a towering creature roaming Ice Age plains. Its DNA forms a faint tapestry in frozen soil, preserved in fragments. Inside that tapestry lies a peptide named mammuthusin, a molecule shaped by eons of evolution. In its original context, it may have helped resist microscopic invaders in the mammoth's body. Now, through deep learning AI, it awakens again, not as flesh and bone, but as a whisper of protection for life today. That eye operates like a patient storyteller, scanning billions of peptide letters, amino acids, searching for patterns that whisper antimicrobial potential. The model, called APEX, reads the extinctome proteomes from 208 ancient species and identifies over 37,000 peptide sequences predicted to fight bacteria. Out of these, more than 11,000 are entirely absent from any living organism nor remembered. In present-day genomes, picture yourself floating above a lab bench, watching scientists synthesize 69 of the most promising peptides. They build amino acid chains using solid-phase chemistry, translating sequence recipes into real molecules. Then, in gentle cell culture and mouse models, they test whether these molecules can combat infections. Many do. Some perform as well. As polymyxin B, a powerful antibiotic of last resort, mammuthusin, pulled from the mammoth genome frozen 4,000 years ago, shows remarkable potency against resistant bacteria in mice compared with today's treatments. Other peptides, aliphasin 2 from straight-tusked elephants, megalocerin 1 from Irish elk, mylodon 2 from giant ground sloths, each emerge from epochs past, carrying unique modes of action never seen in modern antibiotics. You notice how these peptides kill bacteria. They depolarize the inner cell membrane, creating tiny breaches that bacteria cannot resist. This contrasts with most standard peptides that target the outer membrane. It is as though these ancient molecules found an overlooked vulnerability in bacterial structure, honed over Ice Age struggles and vanished worlds. Drifting through this narrative, you feel the rhythms of discovery slow your mind. Ancient life, machine learning, lab synthesis, they connect across time and space, inviting awe and calm. You imagine the AI model reading cryptic sequences, like a river reading contours of stone, recognizing forms that nature once crafted, now lost. The AI's gentle filtering reveals candidates that, when synthesized, may heal modern illness. Float further down a sequence corridor. First Neanderthal-derived peptides, then human microbiome-sourced fragments. In earlier studies, the same team discovered Neanderthalin 1, a molecule encrypted in Neanderthal proteins, and found it effective in mice as an antimicrobial agent. These molecules were hidden inside proteins with other functions neural, cardiac, metabolic, but tucked inside them, encoded in silent letters, lay immunity. Through AI mining, these fragments were revealed and freed as possible medicine. As you listen to this journey, your mind drifts through timelines. Mammoths roaming tundra, Neanderthals by campfires, peptides evolving across eons, and today's lab bench humming with possibility. Each scene is soft-edged, comfortable, yet alive with wonder. The scientific facts are rich, but woven into gentle imagery rather than dense, technical description. You sense the weight of the global challenge. Antibiotic resistance causes over 5 million deaths annually and threatens up to 39 million by 2050 without new treatments. Yet this challenge is met not by frantic speed, but by the careful excavation of ancient molecular wisdom, guided by AI, tested with human compassion in controlled experiments. This chapter closes with you resting on a pillow of knowledge. Ancient peptides reborn through molecular de-extinction. AI as gentle archivist, and hope emerging in the shape of tiny molecules resurrected from deep time. You feel ready to drift toward the next chapter, where deeper learning unfolds, how AI becomes your companion in imaginative creation of entirely new peptides, shaped from patterns yet unseen. You drift into the vast archive of evolution, where fragments of extinct life resonate in your mind. Secret codes in fossil DNA stir softly, hinting at molecules that once protected ancient bodies. AI becomes your gentle guide, unraveling these whispers from deep time. You sense the living archive inside us, and beyond awaiting discovery. You settle deeper into your comfort, eyes closed as the hush of the world around you fades. In this quiet space, your mind opens like an ancient vault door creaking softly. You are not bound by time. Instead, you float across epochs, carrying your calm presence into eras when mammoths roamed, Neanderthals lit fires, and seas teemed with long-lost creatures. In this journey, your mind becomes a time machine. You carry within you a sense of memory, not personal, but genetic. Like pages in a book, each strand of DNA holds a story. The adaptations to cold, the battles with ancient pathogens, the quiet survival in shifting climates. Among those pages lie hidden codes, fragments of proteins, peptides that once whispered defenses in bodies long vanished. AI becomes your interpreter. In the laboratory beyond your mind, a system named APEX reads millions of peptide sequences across extinct species, searching for hints of antimicrobial activity. It identifies over 37,000 peptide fragments predicted to have broad-spectrum effects. Among those, more than 11,000 do not appear in any living organism today. These fragments, once silent, now speak through prediction, guided by deep learning. Imagine the AI as a patient librarian, flipping through volumes of life's history. It reads amino acid patterns as though reading poetry, searching for verses that rhyme with defense. Soft pulses of possibility rise as the model scores sequences, detecting motifs that once shifted bacterial membranes, even in creatures extinct. For thousands of years, you feel the hum of ancient molecules. One is Neanderthalin 1, encoded in Neanderthal proteins, rediscovered and synthesized in the lab, effective against infections in mice models. Another is mammuthusin 2, traced to mammoths, which showed efficacy comparable to last resort. Antibiotics like polymyxin B in preclinical tests. These discoveries drift through your mind like soft chiming bells. They are not merely data points, but echoes of lives that lived. Their existence remained dormant until AI stirred their codes awake. You sense the deep continuity between past and present, Ancient survival strategies repurposed for modern healing. The AI's method is gentle in its power. First, it scans thousands of proteomes, ancient extinctomes, from over 200 species, decoding amino acid sequences between 8 and 50 residues long. Then, it predicts which fragments may act as antimicrobials against modern pathogens. Among the top hits, 69 peptides are synthesized, each tested in vitro and in animals. The success rate, over 50% active candidates, far beyond traditional methods. You imagine the lab bench, sterile yet warm with possibility. Robotic pipettes gently assemble amino acids, chain by chain, into peptides like mammuthusin or Neanderthalin. In Petri dishes and mouse models, these molecules reveal their old powers, breaching bacterial membranes, depolarizing cells in ways unfamiliar to modern antibiotics. Each successful result hums with quiet triumph. Your breath matches the pace of discovery. Slow, deliberate, rhythmic. You breathe in past life's resilience, breathe out new forms of hope. The quiet wonder arises from knowing that extinction does not silence entirely. Genetic remnants persist, awaiting rediscovery. Softly, you reflect on how your own body holds thousands of peptides. Some are ancient, some are new, some evolved for defense, some for metabolism. AI has also mined modern human proteomes, finding thousands of hidden peptides with antimicrobial potential, nestled inside ordinary proteins from eyes, nerves, and heart. It suggests your genome too harbors hidden guardians fragments, repurposed by evolution for multiple roles. This chapter is the bridge between two worlds. First, the ancient archive. Next, the generative frontier. You feel your mind opening to the power of invention, not just rediscovery. For if AI can read ancient codes, might it also imagine entirely new peptides, fresh sequences drawn from patterns yet unseen? As you drift, the rhythm of your breathing mirrors the slow extraction of memory. Inhale ancient resilience. Exhale modern translation. Each breath grounds you. Each thought softens the boundary between eras. In this meditative state, knowledge becomes solace. The vastness of time becomes a cradle. You are gently held by evolution's continuity, from extinct mammoths to living human bodies. You sense that every peptide, every fragment, is a bridge binding past, present, and future in molecular whispers. Your consciousness settles in this intermediary space where memory becomes medicine, where AI connects ancient life to modern healing. You are ready to move deeper with curiosity and calm into the next chapter's unfolding. Generative AI dreams of new medicines, where invention follows discovery in softly glowing harmony. AI flows softly like an ancient sea, reading molecular scripts etched in vanished creatures. It listens to patterns hidden in extinct proteomes, discerning whispers of antibiotic promise. Each decoded peptide becomes a story of hope, returning. From deep time to modern healing, you settle into the rhythm of discovery as AI gently illuminates new paths for medicine. You close your eyes and drift into a vast ocean of possibility, where deep learning moves like a slow, rhythmic tide. In this space, AI reads ancient molecular scripts, surfacing peptides lost to time, molecules once encoded in the proteins of creatures long extinct. You feel yourself carried along gently as each fragment is drawn into awareness. The AI, called APEX, is your patient guide. It scans over 10 million peptide candidates from 208 extinct species, the extinctome, searching for signs of hidden antimicrobial potential. Through an ensemble of deep neural networks, it predicts activity based solely on sequence patterns, guided by knowledge from both public databases and lab-calibrated datasets. Imagine the AI as a composer hearing music in genetic code, trillions of notes processed swiftly, selecting those that hum with life. Out of those millions, It identifies 37,176 peptides predicted to fight bacteria, and more than 11,000 are completely new, not found in any living species. From these, scientists choose 69 peptides to synthesize and test in the real world. Almost 60% of them show activity against dangerous pathogens. Some even equal in strength to last resort antibiotics, like polymyxin B, in tests with mice. Molecules such as mammuthusin 2, megalocerin 1, and Neanderthalin 1 gently fight infections, restoring balance quietly and effectively. These peptides work in subtle ways, by depolarizing bacterial cytoplasmic membranes rather than attacking rigid outer walls. They exploit vulnerabilities that most existing antibiotics do not, revealing modes of action that modern bacteria are not primed to resist. As you follow this narrative, your breath becomes soft and steady. You imagine each peptide rising from buried DNA, synthesized by robotic pipettes, then tested with quiet focus in petri dishes and animal models. Their gentle power against infection feels like a healing breeze washing over the past into the present. This is not science fiction. It is real. The research team at the University of Pennsylvania, led by Cesar de la Fuente, pioneers this molecular de-extinction approach. Their work reshapes discovery by bridging extinct biology with modern medicine, all orchestrated by deep learning. In this soft narrative, numbers become lullabies. You listen to 37,176, 11,035, 69 synthesized, 59% success, not as data points, but as gentle pulses of hope. Each number is a chord in an unfolding symphony, where extinct life meets healing. You sense how vast the ocean of sequence space is. Traditional methods mine known peptides, but Apex dives deeper, into unseen territory. It reveals clusters that standard databases cannot reach, illuminating pockets of molecular diversity shaped by evolutionary paths long closed to observation. The AI flows and pauses on promising candidates. Scientists then synthesize peptides, like Neanderthalin 1 from Neanderthal proteins, mammuthusin 2 from mammoth genome fragments, and peptides from extinct giants like sloths or straight-tusked elephants. These molecules, when delivered in mice, reduce infections by thousands-fold, some equaling the efficacy of polymyxin B in controlled preclinical models. Your mind drifts through these layered scenes, AI reading millions of sequences, robots building peptides, mice healing in a slow, deliberate flow. You feel the continuity between eras, extinct creatures facing ancient bacteria, modern labs reviving their molecular legacy to defend human bodies now. This chapter carries a rhythm, deep learning's method, first scanning vast genetic seas, then surfacing candidate peptides, and finally testing them in the gentle world of biology. Each phase moves like water over pebbles, silent, steady, shaping discovery. In this current of story and science, you hear the whisper of potential. APEX's architecture, attention-based encoders feeding prediction networks, reveals hidden patterns in sequence alone. It does not yet peek into 3D structures, but even so, its predictions bring results beyond classical limits. That you feel calm reassurance. Deep learning is revealing not just known peptides, but expanding chemical space, unlocking avenues unimagined just years ago. The ocean of extinct molecules becomes a reservoir of possibility. Your awareness softens into this meditation. Ancient peptides, modern labs, AI reading memory. Each layer meshes into quiet wonder. The world is vast, but you are cradled in gentle curiosity. You sense that this is only the beginning. First, AI listens and discovers. But what if AI could also imagine? What if it could dream molecules not yet written in history? You feel the transition pressing softly, from finding ancient molecules to creating new ones inviting generative invention next. You encounter mammuthusin 2, a peptide from a woolly mammoth sleeping for millennia. Its ancient sequence pulses with antimicrobial promise. Revived gently by AI, you follow its journey from frozen fossil to lab synthesis, to healing in animal models. In soft reflection, you ponder how extinction can echo hope through molecules. You settle into your breath, slow and grounding, as you drift through deep memory. In the hush of your inner awareness, you encounter Mammuthusin 2, a whisper from a woolly mammoth that lived 4,000 years ago. It lies in frozen code amino acids, preserved in ancient DNA waiting for discovery. The AI model, Apex, scans millions of sequences and lingers on this one. It recognizes whispers of antimicrobial power hidden in the pattern of letters. In a gentle awakening, Mammuthusin 2 stirs from geological sleep, emerging as a candidate to fight modern bacteria. Imagine a fossil buried in Siberian permafrost. Within it lies a fragment of the mammoth's protein, a peptide never before seen by human eyes. The extinct code slumbers in databanks, unreachable until deep learning teaches machines to read across time. APEX finds it among cottages of possibility in an ocean of extinctome. Like a musical motif hidden in orchestral noise, it stands out. Scientists synthesize this peptide by adding amino acids in a precise chain robotic pipettes, building the molecule letter by letter. They place it into petri dishes where bacteria grow. The bacteria falter. Mammuthusin 2 depolarizes their inner membrane, causing collapse from within. In mice with infections, it reduces bacterial loads by thousands-fold. Its effect comparable to polymyxin B, a last resort antibiotic. You visualize the lab bench, soft lights, gentle hum of machines, careful hands guiding pipettes. The peptide moves through the culture medium, meeting bacteria. In infected skin abscesses and thigh infections in mice, it restores balance. The results feel like dawn breaking after a long night. Mammuthusin 2 also shows slower degradation in the body compared to some other peptides, giving it more time to act quietly and effectively before being broken down. That durability makes it promising for further study as a therapeutic dot as your breath drifts with this story. Each inhalation connects you to ancient life. Each exhalation brings the peptide's promise to healing. You sense how evolution shaped its sequence in megafauna, facing unique pathogens in Ice Age conditions. For cold, old infections, they sculpted Mammuthusin 2 for resilience. In mice, the peptide's efficacy isn't just statistical, it's softly visible. Wounds heal, swelling recedes, and infection retreats, not through harsh chemical force, but through calculated molecular diplomacy. It resets bacterial balance. You feel warmth in this transformation. You consider antimicrobial resistance's deep shadow. Over 5 million deaths annually today, projected up to 39 million by 2050 without new treatments. The weight of that knowledge settles in your mind. Yet here is a gentle answer shaped by extinct life. Mammuthusin 2 is not rushed. It is deliberate, measured, shaped by time. Redesigned with precision, soft rhythms stitch the narrative together. Ancient DNA, AI prediction, molecular synthesis, lab testing, animal healing. Each stage pulses in sequence like your heartbeat guiding meditation. You recognize the thread, de-extinction of molecules. Not to revive mammoths, but to resurrect molecules that once fought disease. Mammuthusin 2 belongs to a cohort of peptides. Elaphasin 2 from straight-tusked elephants, Megalocerin 1 from giant elk, Mylodonine 2 from giant sloths, all resurrected and tested in mice with success comparable to polymyxin B. In this moment, you feel awe at the span of time. A creature that strode across Ice Age plains, lends you one of its molecules to heal modern bodies. The ancient becomes intimate. The extinction offers rescue. The dead become allies. Your mind softens. Extinction doesn't erase everything. It hides patterns that endure until we learn to listen. Mammuthusin 2 is a bacterial warrior clothed in ancient memory and current design. Beneath the scientific facts, amino acids, membrane depolarization, mic values lies a poetic resonance. Survival beyond death. The molecule speaks of continuity, of hidden potential in silent archives. It reminds you that extinction isn't final. It's dormant, like seeds waiting for spring. You remain steady, breath slow. The peptides journey begins in Ice Age genetics and flows into modern labs and possibly future medicine. Each step is gentle but determined. Evolution shaped it. AI found it. Humans tested it. As your awareness drifts, the story opens to broader horizons. If AI can resurrect Mammuthusin 2, what else might it find? What beyond ancient memory can it craft? Could it compose entirely new peptides, possibilities that evolution never wrote? You sense the next chapter stirring, from rediscovery to creative invention. The mind that reads can also imagine. You feel prepared to drift into that exploration, into how AI might dream new molecules into being. You enter a gentle lab world where extinct genomes breathe again through careful chemistry. Neanderthal and mammoth peptides awaken softly under AI's guidance. No dinosaurs return, just molecules, revived with intention and human care. Here, you sense hope in hidden codes transformed into healing tools. You rest into the rhythm of your breathing, slow in, slow out, carrying your awareness into a calm lab space. It feels womb-like, softly lit, spacious, quiet. Here, extinct genomes gently unfold across tables and petri dishes. Nothing fantastic like Jurassic Park, only molecules coaxed into life through patience, chemistry, and deep respect. Your mind drifts to fossil DNA drawn from Neanderthal bones and mammoth tusks. From that data, AI gently selects peptide fragments encoded, sequences that once danced in ancient proteins. The process of molecular de-extinction begins not with rewriting history, but with translating hidden whispers into molecules that might heal today. Consider Neanderthalin 1, found within Neanderthal. Proteins that had other functions, perhaps neural or metabolic. Hidden within, a fragment encrypted secret defense. APEX recognizes it and flags it for synthesis. Or think of Mammuthusin 2, encoded in cold-adapted mammoth proteins. Now reborn molecule by molecule, robotic pipettes become your silent teammates. They assemble amino acids in gentle order, chain by chain, as if weaving a garland. Each peptide emerges. Neanderthalin 1, Mammuthusin 2, Eliphasin 2, Myelodonin 2, each born from molecular memory. The process is exacting yet unhurried, like a craftsman threading beads into a necklace. When peptides are ready, they move into petri dishes where bacteria dwell. In some dishes, the bacteria thrive. In others, the revived molecules work there. Quiet power depolarizing cytoplasmic membranes in ways modern antibiotics cannot. The result: infection retreats softly, life restored. Further, these peptides enter animal models. In mice with skin or thigh infections, the molecules reduce bacterial counts by orders of magnitude. Healing isn't abrupt or harsh. It's deliberate, careful. The organisms rest, wounds close, and balance return slowly but surely. Scientists working here hold deep respect. They do not seek to resurrect extinct creatures. Instead, they listen to the echoes of genomes and design just a fraction of what once lived. Ethics advisors weigh in. De-extinction of molecules raises different questions than bringing back animals. There is less risk, but still responsibility questions around patenting, natural versus synthetic boundaries, and our stewardship of nature itself. As you drift, you sense the lab bench as a bridge between past and future. Evolution wrote these peptides in contexts now lost: cold climates, different pathogens, unique life histories. Today, they may offer antibiotic strategies untouched by modern resistance. The molecule mammuthusin 2, for instance, is as effective. In mice is polymyxin B, a last resort antibiotic with a gentler, ancient structure. Yet the work is not without its questions. Stability and safety remain under close study. Many peptides degrade too quickly or may interact unpredictably in humans. The path from petri dish to patient is long clinically. It can take up to 13 years for a compound to reach approval. But AI-supported prediction accelerates early phases, reducing guesswork and speeding discovery. You feel the synergy of human touch and machine learning. The lab is a quiet space of collaboration, human insight, robotic precision, algorithmic selection. Here, extinct molecular codes meet modern chemistry and biology to create something gently new. Breath flows slow and steady. You visualize a peptide traversing a sequence of states, encoded in fossil genome, gives read by AI, gives synthesized by robots, gives tested in cultures, is healing in mice. Each step is measured, calm, and precise. Each step brings molecules one breath closer to purpose. You think about the proteome archive. Around 200 extinct species were mined, yielding over 37,000 predicted antimicrobial peptides. Of those, more than 11,000 are entirely new to life, and 69 were synthesized and tested experimentally. A small fraction, but enough to spark hope. You sense that within these thousands lies potential medicine for resistant infection. You feel the emotional quiet of this process, not triumphalist, but humble. The molecules are not superheroes. They are softly forged in labs, shaped by memory. They remind us that extinction hides patterns, not always final erasure. That evolution writes in layers, and sometimes we can read those layers again. In this space, you sense the movement from scarcity to abundance. Ancient peptides once lost, now rediscovered. Extinct life shaping present-day medicine. There is a poetic justice. Modern humans, whose ancestors perhaps hunted mammoths, now receive molecular gifts from them. Your breathing syncs with the narrative's rhythm. Inhale, ancient genomes. Exhale, lab discovery. Inhale, machine reading. Exhale, peptide healing. You are carried gently through time, science, and soft wonder. This chapter closes in calm reflection. You sense that AI has listened to ancient molecules, and now, through synthesis, they begin to live again. Yet discovery is only the first part. Another horizon awaits. What if AI could move from rediscovery to creation? What if it could imagine molecules not found in nature, new sequences, shaped by potential, not past? You feel the question settle softly in your mind. If molecules can return from extinction, could new ones be born composed by AI with purpose and curiosity? You explore a hidden world within your microbiome, teeming with ancient microbial allies. Small peptides, crafted by microbes in your gut, hold untapped antibiotic potential. Even venom peptides, honed by evolution, illuminate new paths for healing. Together, these secrets suggest the next chapter, AI's creative canvas for discovery. You sink deeper into quiet as you feel the gentle rhythm of life within your own body. Nestled in your gut, thousands of microbial species whisper among themselves, each crafting tiny peptides in a soft chemical lexicon. These molecules carry history, defense, and possibility. Your microbiome is an ecosystem shaped over millennia. Microbes compete, sharing space, nutrients, and territory. They produce small proteins, peptides, that act as their messengers and protectors. These microbial languages include SM or FS, small open reading frames that encode peptides the size of whispers. Researchers at the University of Pennsylvania and collaborators mined over 1,700 gut microbiomes encompassing 444,000 small protein families and identified 323 peptide antibiotics, of which 78 were synthesized and 71% showed antibacterial activity in the lab. One peptide, Prevotellin 2, derived from Prevotella copri, performed as powerfully in mice as polymyxin B in treating deep thigh and skin infections. Imagine these peptides as guardians crafted by microbes, living in your digestive landscape. They patrol quietly, helping keep harmful invaders at bay. Each one is a relic of competition, defense, and microbial survival. AI scanned thousands of sequences until these peptides emerged, promising modern antimicrobial use. Alongside these, scientists found another treasure: venom peptides, forged by spiders, scorpions, and wasps, each shaped by evolution to target life, yet repurposed for healing. A study of 185 venoms revealed diverse antibacterial peptides. From scorpion venom came scorpion-derived peptides like bacteridines, which reduce bacterial growth by altering sodium channels in cell membranes. Spider venom yielded cupienins, small cationic peptides that disrupt membranes in both gram-positive and gram-negative bacteria. Even solitary wasp venom provided latarsins, amphipathic molecules capable of targeting resistant pathogens. Let your mind drift through these stories. Your microbiome weaves codes of resistance and balance, while venom peptides evolved in predatory creatures now lend their power to medicine. Each peptide, whether from a human microbe or an arachnid's venom, emerges as an ally in combating bacterial threats. AI becomes your gentle steward. It reads metadata from gut microbial genomes and venom databases alike. Deep learning algorithms churn through over 1 million candidate peptides, comparing patterns, structures, toxicity, and predicted host compatibility. From this digital ocean, only the most promising peptides rise to synthesis. You visualize robotic systems assembling peptides, microdroplets combining amino acid letters in precise sequence, following AI's design. Then, Petri dishes cradle bacteria. Peptides enter and gently unfold their action, depolarizing bacterial membranes, blocking translation, or disrupting internal function. Observing results, scientists see how Prevotellin 2 restored health in infected mice as effectively as established antibiotics. These peptides often work where traditional antibiotics fail. Venom-derived molecules like latarsins and bacteridines attack bacterial membranes with modes so unique that bacteria currently have few defenses against them. Microbiome-derived peptides, by contrast, may spare beneficial microbes, showing selectivity toward pathogens because they are shaped within microbial ecosystems similar to ours. Your breath remains slow. You sense that your body is not isolated, is a living archive, a biochemical repository of ancient defense systems. Microbes and venoms both reveal that nature's laboratory over billions of generations has produced molecules beyond human imagination. AI helps you retrieve them gently, thoughtfully. There is care in every step: peptide selection, safety screening, toxicity testing. The path from petri dish to patient remains long. Many peptides degrade quickly or may affect human cells if misapplied. But AI helps narrow that path, choosing molecules likely to be stable and safe. This chapter's melody is woven. Inhale microbial memory. Exhale venom potency. Inhale AI reading the hidden codes. Exhale healing whispers entering modern science. You feel the unity of microbial and venom-derived strategies both serving the cause of antibacterial discovery. You sense arithmetic, 323, 78, 71% active, millions scanned, thousands of venom peptides considered. These numbers are not harsh. They are soft pulses of possibility. Echoing gentle science. Your inner peace meets wonder. Your body's own microbiome yields healing peptides, and nature's venoms offer molecules for modern medicine. Both speak of evolution's creativity, long predating human design. AI stands between, translating memory into mission. As your awareness drifts, you feel the tapestry expanding. Microbes, venom, extinct genomes, all sources for microbial allies. But what if AI could do more than uncover? Could it dream of new peptides born from pattern, not nature's archive? You feel curiosity stir softly, from exploration of natural peptides to generative invention. The next chapter awaits. AI dreaming. Entirely new peptides, composed with intention and possible purpose. You enter a realm where AI doesn't just recall, it imagines. It composes peptide shapes nature never wrote, born from mathematical melody. These new molecules emerge in gentle form, brought to life in a pipette's drop. Your mind drifts into a space where invention blossoms alongside biology. You breathe in, letting stillness settle around you. In this quiet space, your awareness drifts into a realm of creation. AI no longer listens to ancient echoes it composes. It dreams of new medicine, sketching sequences that never existed in nature, but carry potential for tender healing. Generative AI steps in as a soft composer, a molecular poet. It draws on patterns learned from thousands of peptides and structures. Large language models, LLMs, graph neural networks, and variational autoencoders form gentle brushes on the canvas of amino acid sequence space. This creative process begins with deep generative models. One such model, CVAE-based, learns a latent space of antimicrobial peptides, compressing their forms into a continuous domain where new variants can be dreamt and sampled. Another approach, using GANs, blends patterns from known peptides to birth novel ones. 24 experimentally validated peptides emerged in one study, each carrying activity against bacteria and viruses alike. Imagine a painter mixing colors until a new shade emerges, a molecule born from possibility, not past. AI, after training, can generate peptide candidates targeted for specific traits: potency, stability, low toxicity. Deep generative models may even include non-natural amino acids, expanding into chemical landscapes humans could never enumerate. Manually. In one real example, AMP, designer and AI system built as a foundation model, created 18 novel antimicrobial peptides within days. Almost all of them showed broad-spectrum activity. 2 performed exceptionally in lab and mouse tests with minimal toxicity. Results showed a 94.4% hit rate, suggesting AI's imagination can rival natural evolution under intention. Another study from Science Direct reveals AI-designed peptides built by a generative pipeline that produced promising candidates against resistant microbes. Evidence that creation may be as powerful as rediscovery. Your mind floats past labs deploying such systems. You see AI-guided pipelines crafting sequences, then robots synthesizing them, as though lowering new seeds into petri dish soil. Those seeds sprout peptides that gently test their power in bacterial cultures. Some peptides form pores and membranes. Others block essential enzymes. Some act with unexpected softness, calming inflammation while clearing infection. APEX-Go, an extension of earlier tools, designs analogs of ancient peptides and achieves 85% hit rates in potency testing, with 72% success in improved antibiotic strength, and can even create multimodal sequences that both enter cells and kill bacteria. You imagine these new peptides as newborn stars, each with its pattern, brightness, and orbit. Some will fade. Others will burn steady, offering balance. In the lab, a sequence designed by AI may outperform natural peptides, more stable in human plasma, less toxic, longer lasting. In silico medicine, generate biomedicines. Latent labs and others use generative AI to design protein structures from scratch, accelerating the pace of drug discovery and precision. You sense a shift from archaeology to artistry. Instead of extracting from the fossil record or microbiome, AI now envisions medicine by design. It blends structure, chemistry, and biological constraints into virtual forms and realizes them through synthesis. Breathing slowly, you feel gentle excitement. You sense responsibility in every design. Researchers screen for toxicity via embedded models and simulations. They check for hemolysis destruction of red blood cells and optimize for selectivity. They refrain from designing biotoxins. Ethical. Oversight stays close as AI composes new molecules. Consider halicin, though a small molecule, rather than peptide discovered by AI, using deep learning from chemical space. It ended drug-resistant infections in mice within 3 days of modeling. This example stands as a reminder. A. Creation can shorten discovery and open uncharted territory. You feel the narrative rhythm. Inhale data, exhale design. Inhale evolutionary memory, exhale generative vision. AI moved from echoing extinct genomes to dreaming new ones. These peptides may fold into new shapes, some amphipathic, some cyclic, some stabilized with non-natural amino acids. PepInV even explores peptides beyond standard building blocks, opening molecular possibilities. A new. The pace of innovation parallels your breath. You imagine 48-day pipelines from design to in vivo validation. Millions of candidate sequences considered. Dozens synthesized and a handful show real promise. 2 peptides in one study reduced bacterial lung infection by 100-fold with low toxicity. Your awareness rests in that space between dreaming and healing. Here, invention meets intention. A peptide is not just chemically calculated, it carries purpose. Yet questions hover softly. Could AI overstep? Could sequences go beyond biology's wisdom into unpredictable territory? That is why dual evaluation, computational and experimental, remains vital. Synthesis and testing in animal models anchor imagination in reality. Your breath continues slow and rhythmic. You imagine future molecules not yet written in any genome. Composed sequences shaped for human benefit, healing infections resistant to any known antibiotic. The horizon opens. If AI can compose molecules from scratch, what might it do next? Could it design peptides that target cancer cells? Or shape molecules to ease inflammation? What other frontiers might generative science unlock? You feel a gentle pull toward what comes next. You sense that from generation springs tailoring, designing peptides for precision, targeted properties, dual functions, reduced waste. You enter a tranquil lab space where revived peptides meet living tests with gentle care. Mammuthusin 2, Neanderthalin 1 and others soothe infection in mice with quiet strength. Each experiment is slow, rhythmic, nurturing life rather than rushing it. You sense promise unfolding from lab bench to living being, softening the path to healing. You breathe in softly and feel a calm anticipation as your awareness settles into the gentle pulse of the lab. Here, peptides resurrected or newly designed are tested in mice, one small creature at a time. The tests are neither dramatic nor rushed. They are slow, deliberate, carrying a nurturing environment where life gently responds to molecular whispers that one of the peptides you know is mammuthusin 2, brought to life from ancient mammoth DNA. In mice with skin abscess or thigh infections, this molecule reduces bacterial load by orders of magnitude comparable in potency to polymyxin. B. A last resort antibiotic, but with a softness shaped by ancient resilience. Neanderthalin 1, derived from Neanderthal proteins, also performs remarkably. In infected mice, it halts microbial proliferation, as if the past whispers back life into present wounds. Imagine mice lying quietly in cages, monitored gently. A peptide enters via injection or topical application. Bacteria retreat from soft tissue. Infected skin heals without harsh disruption. This is not trauma, it is careful restoration. Balance returning like dawn light creeping across the horizon. These peptides share a common mode of action. They disrupt bacterial inner membranes, depolarizing cytoplasmic structure from within. Unlike classical peptides that attack the outer shell, these act in subtle intimacy, bypassing existing bacterial defenses, reaching inside where resistance hasn't evolved. In one study, 5 lead peptides mammuthusin 2, elaphasin 2 from straight-tusked elephants, mylodonine 2 from giant sloths, megalocerin 1 from extinct elk, and hydrodamin 1 from sea cows, all treated mice with skin abscess or thigh infection. Single doses inhibited infections for up to 4 days, easing inflammation and clearing bacteria quietly and effectively. Your awareness drifts through the numbers. Thousands of peptides predicted, 69 synthesized, dozens tested in vitro, a handful selected for mice tests, and among those, several heals. These are soft pulses of hope in a rhythmic chorus. Nature's content, machine's insight, human care. You sense the lab as a sanctuary not of sterile urgency, but of measured attention. Animals are monitored, dosages adjusted, health tracked. Toxicity is checked. Peptides like mammuthusin 2 degrade more slowly in mice than many others, offering dwell time to enact their healing, but still remaining predictable and reversible in physiology. The process is not instantaneous. Days pass between administration and effect, and mice rest. Bacterial colony counts diminish gradually. Swelling subsides. The worm of infection unwinds itself. Each stage is carefully recorded. Weight, temperature, movement, appetite. You imagine a rhythm lab, notebooks, charts, observation, each a soft statement of progress. These are early stages, preclinical promise, not human cure. Many peptides degrade too quickly in humans, may lack necessary stability, or face unexpected IMUNE reactions. Yet the data shows potential. The structure, sequence, and mode of action coalesce into candidates worthy of further development. You consider the emotional weight of antibiotic resistance. Millions die each year, projected to rise. Yet here, in these trials, you sense tiny rays of solution emerging from extinct creatures and high design. It is not magic, it is careful science, grounded in lab culture and cautious optimism. Your breath slows further. You feel initial discovery. In earlier chapters I reading extinct genomes, creating new molecules, now solidify into real-world proof. The peptide goes from concept to culture dish to living model. It is reborn in action, that you envision the lab in sequence. Prediction gives synthesis, gives in vitro testing, gives animal model treatment. The rhythm of research becomes your lullaby, steady, purposeful, unhurried. Each success is small, measured. Each step forward is framed in care. You sense broader questions too. How to scale synthetic production. How to deliver peptides effectively in humans. How to avoid toxicity or immune response. Yet the mice tests reveal fundamental truths. These molecules hold potency and promise. The narrative weaves through time. Ancient DNA resurrected, modern AI predicting, lab chemistry synthesizing, mice healing. It all converges into this nurturing, phase testing in living beings with compassion and precision. Within this flow, you grasp the transformation from memory to medicine. Extinct peptides find purpose. New peptides designed with I may soon join them. Each molecule tested is a whisper of possibility in biomedical evolution. You feel grounded in the idea that discovery is a journey, not a leap. Healing emerges slowly, thoughtfully. Each mouse trial is both end and beginning, a confirmatory step and a doorway to human trials someday. As your awareness drifts, you sense the continuity of care. The same lab that revived ancient peptides will next refine generative designs, optimizing for stability, potency, and safety. The path ahead is about refinement and fine-tuning. You sense the narrative, turning from testing existing candidates to tailoring better ones. The next chapter invites exploration of how AI and chemistry combine to fine-tune peptides. Variants with increased stability, reduced toxicity, targeted activity. Slowly, your breath matches the measured footsteps of science. You feel the quiet promise of molecules tested in living systems, reaching toward potential therapy. You prepare to drift further, from nurturing potential in mice to optimizing molecules for human use, precision guided by generative AI and mindful experimentation. You pause in reverent calm, sensing the weight beneath molecular revival. Questions emerge gently. Who owns tokens of extinct life and why? Patents and stewardship blend under soft philosophical light. You remain open, aware that wonder comes with responsibility. You settle into the hush of reflection, inhaling past and present as one. A single molecule, resurrected from a long-dead genome, carries echoes of life and quietly asks, what do we owe this world now that we can listen? Molecular de-extinction gives potential medicine, yet it ripples far beyond the lab bench. Extinct peptides, such as mammuthusin 2 and Neanderthal 1, speak not only of antimicrobial power, but of forgotten beasts and human cousins lost to time. They reside in a gray realm, no longer natural, but not entirely synthetic. They exist only because we brought them back. And that raises delicate questions. Can extinct molecules be patented? Do we own heritage? Or is it common biological inheritance? In patent law, naturally occurring substances, including genes, are unpatentable. Yet extinct molecules pose a novel twist. They no longer occur in living organisms, yet once did. If we synthesize mammuthusin 2, is that invention or recovery? Legal scholars ponder this quietly. The law is unsettled. The University of Pennsylvania's bioethicists and patent experts are still determining whether these resurrected peptides fall into a legal gray zone, and what that might mean for access and innovation. Imagine someone claiming ownership over a molecular echo of Neanderthal life. What does that mean ethically? Are such molecules part of humanity's shared ancestral heritage? Or does patenting them incentivize further discovery that could save lives? These thoughts swirl softly in your mind. As potential wing. There are broader questions too. Do humans have the right to mine extinct genes for modern gain? One viewpoint holds that resurrecting molecules is different from reviving animals. The risks are smaller, the intent more focused, but philosophical questions remain. Some fear overreach, a sense of dominating nature, even through molecular fragments. Others see acts of stewardship, responsibly reclaiming lost tools for medical good. This is not about dinosaurs returning. It is about molecular fragments, resurrected from fossil data and preserved in lab freezer vials. Yet the emotional weight is real. Gratitude to extinct life, humility before evolution, and care for consequences. These are questions no lab alone can answer. As you breathe, you feel a gentle duality. innovation and responsibility joined in soft balance. The same molecules that may heal infections also ask us to reflect. What is rightful use? Who benefits and who decides? Consider the practical perspective. Without patent protection, companies may hesitate to invest in development. Antibiotic. Research is notoriously underfunded despite urgent need. Could patents on extinct peptides incentivize development? Or might public benefit fare better under non-profit models and open access? Scholars are already debating whether such molecules should live in the ecosystem of public health rather than private enterprise. You imagine warm laboratory lights, algorithms scanning proteomes, peptides gently synthesized, and mice treated. Then, step back to consider broader impact. Because discovery does not end at efficacy. It extends into ethics, policy, and shared human fate. You let your breath soften further, embracing the quiet that follows curiosity. Molecular extinction acts like poetry whispered through time, yet it also demands authorship questions. Who gets to write the story? Who claims the line? Who shares its meaning? These thoughts are not urgent alarms, they are soft companions, inviting reflection rather than panic. You sense the ongoing dialogue among scientists, ethicists, communities, and public health advocates. They ask, does resurrected biology belong to all? To none? To those who discover it? Even as AI breathes life into ancient codes, humans must listen carefully to moral echoes. The question is not just what we can do, but what we should do with molecules born of extinct lives and human ingenuity. That you feel a gentle responsibility unfolding inside you. You observe the laboratory's careful process and see a parallel in ethical stewardship. Just as scientists test safety and avoid toxicity, society must test wisdom and avoid harm. And so the current of narrative carries you onward, from rediscovered molecules to dreamed molecules, from discovery to creation. Yet creation bears weight too. If AI can imagine completely new peptides, what then of responsibility? What ethical reflection must awaken alongside invention? You rest in that unfolding awareness, discovery, design, and the ethical context that must accompany both. You envision a future where ancient peptides gently soothe infections and quiet healing. Resistance retreats as molecules reborn from deep time bring hope. Soft optimism emerges in each lab test, each peptide refined with care. You drift toward sleep, carried by the promise of possibility and renewal. You settle softly into the twilight of this journey, breath slow and rhythmic as dusk nestles around your awareness. In this gentle moment, you imagine a world transformed, softened by molecules resurrected from the archives of time. Ancient peptides, once silent in extinct genomes, now reside in vials and tissue cultures, healing where resistance once stood firm. Think of mammuthusin 2, Neanderthalin 1, and their peptide kinol emerging from extinct species and microbiome. Worlds. Their gentle power against infection feels like a balm over wounds inflicted by bacterial resistance. These molecules do not roar, they whisper, softly restoring balance. In clinical development, peptide-based therapeutics have begun to flourish. As of now, 12 peptide drugs targeting antimicrobial or antifungal activity are FDA approved, and more are in trials. The path from molecule to medicine remains long, yet each successful step carries softness in its promise. Imagine communities where the threat of resistant pathogens recedes. In hospitals and clinics, creams and injections infused with peptides derived from ancient genomes and AI-designed libraries quietly heal infected wounds. Resistance rates drop, recovery times shorten. Antibiotic stewardship becomes gentler. Across the globe, healthcare contexts change. regions once devastated by drug-resistant infections, MRSA, VRE, multi-drug-resistant E. coli now witness fewer fatalities. Peptides sourced from microbiomes, venoms, and extinct lineages rediscovered or designed form an expanding arsenal against superbugs, allowing modern medicine to stay one step ahead. You think of nature's microbial equilibrium. These peptides, whether reborn from long-lost creatures or dreamt by AI, are not synthetic chemical weapons. They are evolved or evolution-inspired balancing agents. They disrupt bacterial membranes with precision, rarely leaving resistant survivors. Facing bacteria shaped by nature, these peptides act with evolutionary finesse. Your mind drifts through stories of global collaboration. Researchers share peptide designs openly. Nonprofits and public benefit firms carry them into trials, prioritizing access and stewardship over exclusivity. Some peptides may remain unpatented gifts to humanity, others licensed for broader distribution with affordable pricing. The conversation between inventors and society continues, guided by shared care. As we pondered in ethics, communities in low-resource areas gain access to topical cream or injections infused with peptides, like Prevotellin-2, bactridines, or novel AI-designed molecules. They don't need high-tech labs to benefit. They receive locally produced treatments for skin or soft tissue infections, delivered gently and effectively. You sense the pipeline accelerating. Generative models like AMP Designer create new peptides in days 18 in one study, with 94.4% positive activity rate, minimal toxicity, and efficacy in mice. Some candidates reduce bacterial lung infection nearly 100-fold, all within 48 days from design to validation. The future feels both immediate and sustainable. From ancient retrieval to future invention, the bridge spans eras. Time dilation between Ice Age genetics and present-day healing grows shorter. Peptide libraries expand. Research networks interlace globally. Health outcomes shift, resistance curves flatten. Deadly pathogens lose their strongholds. You breathe in reflection. Life's encoded legacy resurfaces gently through synthesis and stewardship. The silent archive of extinct lives becomes a quiet medicine. Nature's diversity, once hidden in microbial competition, venoms, amphibian skin, or permafrost, now blossoms into healing. The world softens. Infection no longer carries dread, but expectation of recovery. Resistance loosens its grip as bacteria meet unfamiliar molecular patterns. Survival whispers in the link between long-dead creatures and living patients, made possible by AI-guided science. As your consciousness drifts toward rest, you sense a lullaby in this narrative. Peptide doctors' tiny sequences compose healing symphonies in tissues and cells. Their notes are quiet, effective, and lasting. Hope returns in measured doses. Healing is an act of translation between ancient DNA, human need, and peaceful science. Tonight, you rest with that hopeful vision. A world balanced by peptides, born out of time, reshaped by knowledge, intended for gentle healing. And in that peace, you sense one last question. If peptides can heal infection and resistance retreats, might future designs address other challenges like inflammation, cancer, or metabolic balance? Sleep deepens as that quiet curiosity rises, not urgency, but gentle invitation.